for invitation for this conference and nice introduction. Today I want to present some data about determination of the cause of male infertility using transcriptome. But nowadays more and more couples have problem to get child. So how we can define infertility? Infertility we can define as the lack of conception following of frequent unprotected sexual intercourse during 1 year if the couple is under 35 years old, and then this older, this is only 6 months, and approximately affects 15% of reproductive couples, and the ratio is increasing with the age. And what is important, men and women are equally also affected. The internal causes of male infertility are alcohol, drugs, tobacco, some health problems, too much radiation or chemotherapy, and also some environmental factors like too much pesticides, hormones in meal or lead, and also high temperature. In diagnosis, the most of male infertility causes are due to low sperm counts or poor sperm quality. The remaining causes of male infertility can be anatomical problems, hormonal imbalances, or genetic defects. In this year, European Association of Urology showed that 30 to 30% of male infertility is still idiopathic. So, determination of the genes which are involved in spermatogenesis control might help to better understand molecular pathways driving male gamete production and consequently the reasons of male infertility. In our institute, during several years, there were created and analyzed knockout mice models for several genes which were essential for spermatogenesis and then for reproduction. But we detected also several non-essential genes, and we work more with these genes. These genes are specifically expressed only in germ cells during spermatogenesis. It is histone cluster 1, which replaces histone in the first spermatocyte stage. Testicular haploid express gene, which is regulated from some factors from Sertoli cells. CAMP responsive element binding protein 3 like 4, which is active in response to the stress condition. And transition protein 2 is also replacement of histones during remodeling the spermatids. And testis express protein 22 is responsible for biogenesis of acrosome and midpiece in the last step of spermatogenesis. And last gene, acrosin propeptide, which is important for good acrosome construction and then during fertilization and acrosome process. In our institute, all these genes were knockouted, but in single knockout, like and double, triple, and more knockouts in different combination were every time fertile. But when we knockouted 6 genes in some animals where we observed infertility, and it was not problem because we have knockouted all the 6 genes, but it was some other problems, and it was our question how only some % of these animals are infertile. So we analyzed the phenotype of the 6 times knockouted mice, and determine the infertility causes with also analyzes new genes which we obtain from transcriptome. So the first shortly about the phenotype which we received. We observed that about 20% males are infertile. In this time we also in these males we observed all stages of spermatogenesis. So spermatozoa were produced standard like normal, because it's known that serum testosterone level also influence on fertility. We check the serum testosterone level and also total sperm number, and was similar like in this fertile knockouted mice and infertile wild-type mice. But we observed that the sperm speed from these infertile males and also acrosome reaction is destroyed, and also we observed increased number of sperm with abnormal head. Here you can see the how looks like the normal, and in different way destruction the normal shape of head. So because it's known that the transcriptome tool we can obtain some clusters of co-regulated genes to the process, clusters of genes involved in the same biological process, or cluster of genes expressed in the same organelle. We decided to compare transcriptome, these fertile and infertile males. We observe because it was also from literature known that about 2,700 novel transcripts were identified from these mouse germ cells like cytochrome C, which regulate apoptosis in germ cells, or another factors which regulate which are transcriptor regulator during spermatogenesis. We use a gene chip mouse array from Affymetrix. It was to some small plates, and we compared this fertile and infertile knockout mice. On this plate we tested 21,000 genes, and we obtained 55 genes which were down or upregulated between these fertile and infertile males, and from them 41 genes have known expression in testes. 4 were testis specific. 4 genes were completely unknown. There was only short sequence obtained from some computational predicted database, and 10 genes which were known from other organs than testes. But for most of them, the expression in testes were not tested. So the first we tested these 10 genes, but only one of them was in testes specific only. But the most interesting was this gene. It was predicted gene. It was short gene, which was the strongest down-regulated between these fertile and infertile males. So we decided to analyze more detail this gene. When we confirmed this transcriptome via real-time PCR, we analyzed this gene, and in different infertile 6 times knockout males, the expression was not present, whereas in infertile was strong present. And also by quantitative PCR, we didn't see this expression in infertile animals. Then We analyzed the expression of this gene in different organelles and was expressed in testes, and epididymis was really strong reduced, and another organs was not expressed. So we concluded that it might be interesting gene for fertility. Then we analyzed the expression of gene in different days postpartum in testes, and expression start in 16 days postpartum when primary spermatocytes are produced. This we analyze also in different cDNA obtained from mutated males mouse with mutation in with spermatogenesis arrested in spermatocyte, first primary spermatocyte, secondary spermatocyte, and spermatids, and were present. So these results confirm our results before. It was not expressed in testes and epididymis in mice with completely arrested spermatogenesis, and was also not expressed in Leydig and Sertoli cells. So we conclude that this gene is strictly expressed in germ cells. To be sure that this gene is expressed also in spermatozoa, we isolate sperms after mating, male and female, from vaginal plaque positive female, and we isolate and obtain this gene. As a control in uterus, it was not expressed. Also, epididymis in 20 days postpartum was also not active. We also prepare castration of these males, and 3 weeks after castration we couldn't observe any expression in epididymis. So it suggests that this RNA might be stored in spermatozoa and useful. It's known from literature that mRNA might play a certain role in fertilization and embryonic development during different reorganization of DNA, formation of pronuclei, or activation of oocyte. It's also known from establishing the imprinting in early embryos. There is also some iRNA which inhibit genes active exclusively during embryonic development. Then we wanted to know if it's important to fertilization and developing this fertilized oocyte during early embryogenesis. We mated males with superovulated females, and 48 hours later after this mating, we checked the two-cell stage. This is the first embryonic step, and we observed that from fertile 6 times knockout, we observed this two-cell stage in 35%, but from infertile it was nothing. So the next question was, is active this? Is it possible to fertilize or not? So to find the best time for isolation to pronuclease stage from fertilized oocyte, we try in different time points after fertilization, and from wild type, every time we receive about 20% to pronuclease stage, but from fertile it was every time about 3–5%. And in the last this last hours we received also 2-cell stage already. So it was not possible to establish good time to to find these two pronuclei from potentially find this two pronuclei stage from infertile males. So we conclude that this time depends on fertilization time and takes place during 48 hours after mating. You never know when when directly the mating will be, the contact will be done. So to answer our question, we create some in vitro fertilization experiment. And the first, it was in vitro experiment with oocyte, where we used oocyte with zona pellucida, with complete oocyte. And here we observed a reduction from between fertile and wild type in two cells stage. So in infertile we didn't observe this two cell stage. So then we destroyed the zona pellucida using thyroid acid, and we could observe a similar percent of two pronuclei and one cell stage in all three groups: wild type, 6 times knockout fertile, and infertile males. So according to all our in vitro experimentation, it seems to be that the sperm from infertile males are not able to penetrate the zona pellucida to intact oocyte. So then we concentrated on this gene again and check and compare the sequence of protein with database. We find that this gene encodes a protein which is similar to capping protein alpha subunit family, and members of this protein family regulate growth of the actin filaments by capping the barbed end and growing actin filaments. In normal cells we have actin cytoskeleton, and this capping protein blocks polymerization and depolymerization this actin. So, and as we know, the actin is important to create the cell shape. It's important also in cell division, and the polymerization to allow the activation of the outer acrosomal membrane during acrosomal reaction, and prevents the sperm from incorporation into oocyte cytoplasm. It's also important in maturation of sperm and can block sperm motility. We also we also create antibody against a specific epitope in this protein, and this we observed expression. We observed the protein in testes, and then we also prepare experiment of colocalization this protein with actin capping protein subunit alpha antibody, and this antibody, as you can see here, it's nearly it's nearly 100% colocalization with this new gene, new protein. So we suggest that our gene might be a new protein, might encode new protein, which is new capping protein. So our conclusion, what might be the reason of infertility? We suggest that dysregulation of germ cells, actin cytoskeleton reorganization, might be the cause of infertility in these males with 6 knockouted genes, because this gene the protein encoded by this gene might be important for the regulation of the actin cytoskeleton. We observe increased number of abnormal head. We observe also reduced acrosome reaction and reduced sperm of reduced speed of sperm, and the problems with fertilization. Finally, I want to say thank you for Professor Wolfgang Engel, the head of Institute of Human Genetics in Göttingen, and also to all my colleagues from this university. So thank you for your attention.